All right, welcome. With this video, we want to introduce the functional unit of the muscle, which is a sarcomere. But to do that, we've got to first take kind of a global view and see what we're talking about, where we're at in this big picture. So let's start over here by labeling what we should all recognize as a bone. And the way movement works, right? I've got muscles. They attach to bone through tendon, and as the muscle contracts, it pulls on that tendon, which pulls the bone, and that gives us movement. So this first section here is going to be all tendon. And I want you just to appreciate, we'll come back to this in a minute, but I want you to appreciate that within that tendon, we've got all of our colors. We've got the red on the outside, we've got the blue underneath that, and then we've got the green. So let's go through and let's describe these colors, these separations within the muscle, and help us understand how this whole thing is arranged. So the muscle itself is surrounded by what we see here in red. This is the epimysium. Epi meaning on top of or around the muscle. And this is also what we call the fascia. This is essentially the wrap that goes around holding the muscle together. Now, within the epimysium, we see these blue structures. These are called fascicles. And the fascicle is essentially a bundle of muscle fibers. So by default, then, these green structures are the muscle fibers. And when we say muscle fiber, what we're referring to are individual muscle cells. Now, I think it's important just to take a step back and recognize that when we say muscle cell in the adult, these cells are huge, right? That means that, looking at my quadricep, I've got a muscle fiber running from beginning to end that can easily be measured at over a foot long. These cells can be very, very big, but they weren't born that way. During embryonic development, they were born as individual normal-sized cells, but during the process of muscle development, these muscles fuse together. That's why when we think of skeletal muscle, one of the unique things about them is that they're multinucleated. They have lots of nuclei because they are the fusion products, the syncytium, of lots and lots and lots of embryonic myocytes. Regardless, that muscle fiber consists of a single muscle cell, multinucleated and very large in some cases. Well, now that we understand that this green thing that we're looking at here is an individual muscle cell, it's important to recognize that it's wrapped by connective tissue, very similar to these other structures. We saw with the biggest structure, the epimysium, it's wrapped in that epimysium, right? The fascia. The fascicle itself is wrapped in connective tissue called perimysium, and the muscle fiber is wrapped in connective tissue called endomysium. Now it makes sense. If we go back and look at the tendon, all of these connective tissues, the endomysium, the perimysium, the epimysium, they're all continuous throughout the length of the muscle. And when the muscle itself ends, this connective tissue continues. And what do we end up with? We end up with the tendon, which is just all of the endomysium, perimysium, and epimysium compacted together, where it then attaches to the bone. Well, if this is what we're pulling on, then let's see what we need to attach the actual muscle cell to the connective tissue. So in here in pink, this is the plasma membrane of the muscle cell itself. We call this the sarcolemma. And again, this is just the plasma membrane, a typical lipid bilayer like we have on other cells. Within that, we see multiple subunits called myofibrils. And these myofibrils, as we can see here, can be visually divided up into segments that we see right here from Z disc to Z disc. This segment is called a sarcomere. And if we take a cross-section of this, we see that this consists of really two types of fibers, what we call thick filaments, and these others that we call thin filaments. And we'll talk more about those in just a second. The sarcomere is the functional unit of the muscle. So when we think about muscles contracting, it's the sarcomere that's actually contracting. So, functional unit. Now we can see with these individual myofibrils made up of sarcomeres, these are what's contracting. This is how we're going to connect the muscle itself, the muscle cell, to the outer connective tissue, in this case, the endomysium. And we see that through this structure right here. There's actually multiple proteins in here drawn in yellow, but the primary one that we want to just mention is called dystrophin. And this is the protein that's actually mutated, genetically mutated in the inherited disease Duchenne muscular dystrophy. Without this dystrophin, the muscle can't connect directly to the connective tissue, and so even though the muscle might be contracting, there's nothing to grab onto. There's nothing to adhere to, and so I don't get the movement that I want. And over time, the muscle atrophies because there's no resistance. You lose all your movement, and without treatment, it leads to an early death. Well, with the basic anatomy of the muscle now taken care of, what we really, really want to explore further is this sarcomere, to understand how this acts as the functional unit of our muscles. So as we saw in the previous picture, the boundaries of one individual sarcomere are called Z-discs. So here's our Z-disc, and from one Z-disc to the other, that represents one sarcomere. Now the main proteins that we want to focus on, the main structures, the anatomy of the sarcomere, these massive blue lines, we call these the thick filaments. And we'll take a closer look at these in just a minute. Coming in from both sides, here in yellow, this is the thin filament. And like I said, there's lots we could talk about, but I think the other really important thing that we want to show is this protein here. It's not really shaped like this, but I draw it that way so that we can understand its function. This is titin, and titin's job is to support or attach the thick filament to the Z-discs. And I draw it like a spring because it's important that that can move. And we'll try and highlight that in just a minute. When muscles contract, the Z-discs themselves will move closer to each other. So this Z-disc is going to move in this direction. This Z-disc is going to move in this direction. And we can actually compress the overall width of the Z-disc by about 50%, which is a significant amount. Before we talk about exactly how that happens, there are a couple of anatomical features that we do need to mention. If I was to look at this under a microscope, talking about histology for just a second, there are certain boundaries that we need to make sure we see. Number one, if we were to look at this gap right here, which is essentially measures the overall length of the myosin thick filament, this is what we call the dark band or the A band. A meaning anisotropic. It won't move, and we'll discuss that in just a minute. Another boundary, and to see this one clearly, we need to expand our sarcomeres, because this boundary crosses sarcomeres. So let's draw that neighboring sarcomere. If we were to look at this gap, essentially in between sarcomeres, crossing the Z disk, this is what we call the light band or the I band. And then lastly, there's one more. Well, I guess we could throw in, we already labeled the Z disk, right, running down the middle. This is the M line, or median for middle. And the last one we want to look at is the distance from one thin filament to another thin filament. And this is called the H zone. H for heller, German, meaning bright. So these are all things anatomically that I can see under a microscope. What I want you to appreciate for just a second is we talked about how the sarcomere, during contraction, the Z disks are going to move closer to each other. Think for just a second about what's going to change as these Z disks approximate each other. I hope you can appreciate that the dark band does not change. Right? The dark band is made of thin filaments. They're not changing in any way. So dark, we'll draw delta for change. This does not. However, if you can appreciate, as the Z disks themselves move closer to the boundary of the dark band, this gap is going to shrink. And so this overall light band is going to shrink significantly. So the light band does change shape with contraction. And also with the H zone, right? The whole idea is that these thin filaments are moving in this direction. These thin filaments are moving in this direction, both medially toward the M line. And because of that, the H zone is also something that will change shape during contraction. All right, now let's take a quick close-up look at the thick and thin filaments. Let's start with the thick filament. The thick filament itself is made up of multiple proteins that we call myosin. And myosin, the easiest way to think about myosin is to think about a golf club. So it's got a long shaft on it, and then it's got a head. We can label some of these as, I guess, the couple of important things. This is the hinge region, because as hinges do, this bends. And this part of the head here, this part will bind to actin. And we'll show where that actin is in a minute. Of course, the thick filament itself isn't just one myosin, it's multiple myosins. And they're oriented all around. But please notice, the heads themselves are only at the terminal end, right? This would be the mid line, and we would have, for the rest of the thick filament, these would also be extending outwards like this, same kind of thing. But that's all it is. It's myosin, a bunch of myosins, and with each myosin, I have at the terminal end of it, lateral to the midline, I have these myosin heads on a hinge region that can flex and bend, and the heads themselves bind to actin. So this is the thick filament. All right, let's take a look at the thin filament now and see if we can start to understand how all this stuff works together. So the thin filament, maybe this is coming over here and it's attached to this Z disc here like this. What happens if we zoom in and take a look at that? Well, what we're going to see, actually, we're going to see a helix, two helical strands wrapped around each other. And if we look at these strands closely, let's zoom in and see what this strand looks like. The strand itself is made up of individual little ball-like structures. Each of these is called a G actin. G stands for globular actin. When I string them together like this, like pearls on a necklace, the string itself is called F-actin, which stands for filamentous actin. It's all of these G-actin monomers attached to each other, essentially glued together. A couple of things I want to point out. Each one of these G-actin monomers has on it a binding site for myosin. Remember, we said just a minute ago that the myosin head binds to actin. Each one of these G-actin monomers has a binding site for actin, or for myosin. However, if I want to relax the muscle, I've got to prevent that binding. So we have another player. This is still part of the thin filament. This is called tropomyosin. And tropomyosin has one job: to hide the binding sites so that myosin cannot attach to actin. Now, the third component that we want to talk about, there's several others, nebulin included, but the one that we really care about next is troponin. Troponin is a heterotrimeric protein. In other words, it's got 3 different subunits to it. This is troponin. And you can see the 3 different things that the 3 different subunits that I've drawn. This first one is called troponin I, and this has the job of binding to actin. This next one is called troponin T, and this one binds tropomyosin. And so we can see that troponin is kind of the thing that holds all of these structures together. This last one here, this is called troponin C. And troponin C has the important job of binding calcium. Now, we haven't talked about calcium yet, but now we're going to begin to understand how the muscle actually contracts. In the presence of calcium, what does calcium do? Calcium, when it binds to troponin, specifically, we're binding to that troponin C, right? When calcium binds to troponin, it induces a conformational shape change. In other words, calcium causes troponin to change shape. And this has a very, very important effect. This causes tropomyosin to move out of the way, and it exposes those binding sites. So, in the presence of calcium, what might this look like? Right? We've got all of our G-actin monomers held together. So this is F-actin. We've got all of our binding sites. And tropomyosin was holding those, or basically hiding them, because it was covering them. But when calcium comes in, and calcium binds to troponin, so here comes calcium. These troponins are about every other 7. So put another one down here. As calcium binds, the tropomyosin is lifted off and moves out of the way. And what that allows is for the head of our myosin to come in and now bind. And that's what initiates cross-bridge formation. And we'll spend another video talking about that. But that will lead to muscle contraction. So, in summary, if we zoom out and look at the big picture, we recognize that the sarcomere is the functional unit of the muscle. It's made up of Z discs that are the boundaries, and then thick and thin filaments are the most important components. The thick filament itself is the myosin. It's made up of multiple myosins. Each of them have heads at the terminal end that bind to actin. The thin filament is made up of 2 strands of F-actin. The F-actin itself is composed of individual G-actin monomers. Covering all of the binding sites on those G-actin monomers are tropomyosin, and then troponin is what holds it all together. To initiate muscle contraction, calcium is allowed to bind to troponin. And we'll talk about those mechanisms when we do neuromuscular junction. As calcium binds to troponin, it forces a conformational shape change in troponin, causing it to lift tropomyosin out of the way. That exposes the binding sites on the individual G-actin monomers, allowing myosin head to bind to actin. And when it binds, that initiates a change in myosin where the head ratchets forward. It pulls forward. And that's the movement that we want as myosin works to pull the Z disc in medially toward the midline. That's muscle contraction.